In the fifth lecture, Biotechnology Principles and Processes, let us continue with uh, the vectors, cloning vectors. Now, if you can recall, in the fourth lecture, I have already talked about cloning vectors, plasmids, PBR322, what are the characteristics and how they are used. Now, therein we talked about markers, selectable markers, where antibiotic resistance gene is used as a selectable marker. Now, to use the antibiotic resistance gene as a marker often creates some problem in the sense that it is quite time taking and also cumbersome, like you have to first plate it in one medium, put it in ampicillin containing medium, then again put it in tetracycline containing medium as stated in the example. So it takes quite a lot of time and it becomes little hassle. So to make it hassle free, later another type of marker was found out. Now that process is known as insertional inactivation. Now what is this insertional inactivation? Now what is done? The vector, okay, the vector that is used here also you can see it is a plasmid. Now this vector that is selected is such that it has the gene LAC-Z gene. For example, okay, LAC-Z gene is a gene which produces or which helps in transcription and translation of galactosidase. Galactosidase is an enzyme. Okay, it is an enzyme which is actually used by bacteria like Escherichia coli to digest lactose. To digest lactose, galactosidase is used. Now, this galactosidase enzyme is coded by the gene LAC-Z gene. Now, a plasmid is selected or a vector is selected such that it will have this gene. This gene, it undergoes transcription producing mRNA and then translation forming the functional LAC-Z protein. LAC-Z protein here is galactosidase. Okay, now this galactosidase, when it is produced, okay, the bacteria which will contain it, they will definitely produce this particular type of protein enzyme. That enzyme, when it reacts with a chromosonic substrate, okay, chromosonic substrate is something which is used to mark out or which gives colors on reacting with some other compounds. So when LAC-Z gene, when chromosonic substrate is added to anything that will contain that galactosidase, it gives blue colonies, blue coloration. So if you are getting a blue coloration, it indicates that those bacteria are producing galactosidase. If it does not give blue colonies, that means here you can see it is enlarged here. Some this white, these are not giving blue colonies, whereas the blue ones are giving the these are the blue ones. The blue ones indicates that they are producing galactosidase. The white ones indicates that these bacteria are not producing galactosidase. Okay, now what happens, what is done here when we are trying to use it as a vector? What will be done? The new gene that we want to insert, okay, that will be inserted in the portion which was carrying LAC-Z gene. So using restriction enzyme, LAC-Z gene containing region will be cut out from the plasmid and to it will be added the new gene. So here you can see to it will be added the new gene, the insert. So when it undergoes transcription, okay, when it undergoes transcription, it will not produce the, and also translation, it will not produce the galactosidase enzyme or the protein which was earlier producing, because it had the gene, here it was replaced. So now what will be done? It is not no more producing that particular bacteria. Now this new recombinant, this has become a recombinant DNA. Now this recombinant DNA will now be inserted into a particular bacteria. When it is inserted, it will no more produce it will no more produce the galactosidase due to the absence of the gene, and accordingly also it will never produce the blue colored colonies. So when you are plating it for the first time itself, if they are giving white colonies, means what? Gene has been removed, original gene has been removed, and it has been replaced by a new gene. So that will indicate that yes, this particular recombinant DNA has become a recombinant DNA. It has taken up our new insert. If it continues to give blue color, means the new insert was never added up. Okay, so that is how we can find out whether the new gene, new DNA, the foreign DNA got added up upon the plasmid or not. That is how they can act as a marker. Okay, so here this technique is known as insertional inactivation. That means we insert it and thereby inactivate the the production of the original protein. So here again the same thing. Okay, once more it is shown here. So foreign DNA insert, this is echo R1 sites. Okay, this is the disrupted, that means the Z gene is removed and the foreign DNA has been placed here. Transformation, foreign DNA enters the host cell. So here the whole thing is made to enter into the cells here. Then selecting cells that have received a plasmid. So now when so here what will be done, we will let them grow in the plates. Okay, these particular bacteria will be allowed to grow in the medium. So distinguishes cells carrying insert containing recombinant molecules from cells carrying vectors without insert. So if they are giving the blue pigment, blue color, that means the insert was never added upon. But if they are giving no blue colonies, okay, no blue colors, that means the vector, the new gene got inserted into the bacteria, okay, via the vector. So this is also presence of blue color. Showing of blue color means the transformation did not take place, and if no blue color, the transformation took place. So this is insertional inactivation. Now next we will go to vectors in plants. Okay, now what are the or how do vectors work in plants? Now, as already I had talked in my previous lecture, that in vectors in plants are the Agrobacterium tumefaciens. Agrobacterium tumefaciens is a bacteria which is also called as natural genetic engineer. Why is it called as natural genetic engineer? Because this Agrobacterium tumefaciens, this is a bacteria. Okay, this is a bacteria. It has a plasmid. Definitely it will have a plasmid. It has a plasmid, and in the plasmid, okay, there is a portion. There is a gene portion. That is, if this bacteria ever infects a plant, okay, it enters a plant, then this plasmid has a very special ability, okay, that once it enters into the plants, this plasmid, the DNA of it can go and link up with the DNA of the plant. Okay, it has the special ability. Here you can see the tumor inducing genes have, okay, so this is like when we are doing for a recombinant thing. So the tumor inducing gene, it is called as tumor inducing gene. So these genes will go and manipulate, adapt with the DNA of the plant cell, and thereby induce the plant cell to undergo unlimited growth at the particular cell or particular tissue wherever that gene has entered. Gene of the agrobacterium, which we are calling as TI, tumor inducing gene. So that now what level? Consequently, that portion of the plant will continue to grow in an unlimited, uncontrolled manner, producing a tumor, an outgrowth. Okay, it is actually affecting the plant. Thereby it's a disease causing bacteria. Now that very property was exploited. So what is done here? First, we take out the plasmid from agrobacterium, the TI plasmid. Okay, so the gene, okay, the gene which causes, okay, so that in the restriction site, the plasmid, it is we're using the restriction enzyme. The insert is added here. What is the insert? The insert is the foreign DNA that we want to place in the plant. Okay, so we want to place in the plant. So that foreign DNA here, this is the red is the foreign DNA, is inserted into the plasmid. This portion, see here, it is inserted here, thereby we are creating the recombinant TR plasmid. Already we have seen how it can be done using restriction enzyme and DNA ligase. Then after producing it, this recombinant plasmid is induced into a plant cell, introduced into the plant cell. Now to introduce into the plant cell, definitely it is again placed back into the agrobacterium, because only the plasmid cannot enter into the plant cell. We have to make it enter into plant cell through the bacteria. So this plasmid is again inserted into the agrobacterium. Transformation that we call as, it is made to enter into the agrobacterium, and then the agrobacterium let it attack the plant. We will place it in the plant cell. Okay, letting the bacteria put it into the plant cell. So here we have put it into the plant cell. Now the DNA carrying the new gene is present along with the agrobacterium. So here agrobacterium has this property of transferring its DNA to the plant DNA. So what will happen? The DNA, this foreign DNA, will be transferred into the plant DNA. So now what will happen? The plant cell is getting the new gene. Okay, the plant cell has got the new gene. How did it get the new gene? Because this agrobacterium itself has the property of transferring its DNA to the plants. Okay, which otherwise causes disease. But now what have we done? We have removed the disease causing property. Disease causing gene has been removed and has been replaced with a new DNA that we want the plant to produce. So what will happen? It will become a part of the plant cell, and the plant cell which grows into the new plant, it will have the new trait. Whatever trait we are desiring, it will be having that. Next, we will talk about vectors in animals. Now, in animals, the vector that is used is retrovirus. Now, retroviruses are viruses which have RNA as a genetic material, but it has a special type of enzyme called as reverse transcriptase, whereby it can convert, or it can form the it can form a DNA which will in turn go and link up with the animal DNA. The cells, animal cells that contain the DNA, it can go and link up and accordingly carry out transcription and translation as like the animal. Okay, as like the animal DNA. So thereby produce the property or the trait in the animal. So here what is done? Okay, the vectors in animals, in what is done is, first the cells are harvested from the patient. Patient means here this therapy, this is usually used as a therapy, that means to treat any diseases, because in animals we usually don't desire a new trait. Okay, in some animals it is desired, but mostly in humans, here we are taking the example of humans, it is usually used to as a treatment for some diseases, genetic disorders. So what is done? The cells which has a defective gene, we want to replace the defective gene with a functional gene. So what will be done? The cells are harvested. The defective cells are taken out from the patient. Okay. Then after that, in the lab already the retrovirus is altered so it cannot reproduce. So in the lab what is done? The virus is manipulated in such a manner that it will not produce multiple copies inside the animal, otherwise it might cause some disease. So that way it is modified. Then after modifying, what is done? The gene that we want to place inside the human is added into the virus. Okay, the gene that we want to place in the animal is added to the viral genetic material. Now that viral genetic material, okay, now the whole virus, that means the altered virus is mixed with the patient's cell. Okay, so now the cells that were taken out, into them the virus is allowed to enter. Okay, allowed to react. Now what will happen? The cells become genetically altered. As already I told you, the retroviruses, the property itself is present. Naturally that property is present, that they can make their own genetic material link up with the animal's genetic material, the cell's genetic material. Same way as it happens in the plant. Same way it happens here also. So it becomes, the cells thereby become genetically altered. Okay, genetically, of course, the care is taken that the particular gene we want to replace only that part, or even if that part remains, no problem, because it is already non-functional, we have added up the functional part. Now these altered cells are then placed back into the patient. Okay, and then in the body, now what will happen, it has the functional DNA, so that functional DNA will produce the desired protein which was not being produced earlier in case of diseases. But if it is a case of, like, say, it's a case of we want to add up some new character, new trait, produce a new protein in some animal, generally it's done in animals, that thing, not in humans, then also the same thing, we add up the new DNA, the DNA will produce the desired protein. It is expected. Okay, it will go on in that manner. So this is how vectors in animals work. So what are the four types of vectors? They are bacteriophages, they are plasmids used in microorganisms, and in plants agrobacterium tumefaciens, and in animals retroviruses. So next we will move on, the next lecture we will move on to vectorless gene transfer. That means some gene can also be transferred without using vectors, and then we will also learn about how the cells into which we want to place the vector can be made competent. That means they can be they have to be created such that they accept the vector, such that they accept the new gene. So that will be talked about in our next lecture.